Wow, okay. All right, good morning, everyone. Couple announcements before we get into content. Number one, for anyone that's in the room and also took the exam, exam 2 or exam 1, they're in the room for the last time. If you do not pick them up, just swing by my office in Biodesign C 279, and you can pick them up anytime. In the event that you come to Biodesign, the Biodesign building, and they tell you that you need someone to escort you upstairs, just tell them you're coming to pick up an exam. I've told them to allow you to do that. I wanted to just mention something about sort of the exam grades and everything. Okay, so first, actually, exam 1, exam 2, both of them have now their keys on the module section, as well as the regrade petition form. So in the event that, for example, there's a miscount, or if there's something you want us to take a look at again, feel free to put in the regrade request form, but it has to go through Abesh first. Abesh is going to give it the first scan, and then he'll send it along to me if he feels like it's warranted. He does a pretty good job of doing that. One thing I would say is I definitely try at least to be as lenient as possible. I'm in a, I want you guys to do well. But there are situations where the miscounting has happened, so feel free to send that along. Or if you want some further clarification after Abesh bounces it, if he does, then just let me know. I'd be happy to talk to you about that. You guys are doing exceptionally well, I would say, especially on this last exam. It was really great. I'll have some. We should be about on pace, I think, for exam 3 to be not next Monday, but the following Monday, to give you guys to hand it out on that Monday, and then it'll be due in a week. But we'll have a little more information on that in the upcoming class. I'm gonna give you a good outline of what the rest of the semester is gonna look like. And at the end of class today, just make sure everyone got this message. I'd like to just meet with the honors and graduate folks just to see where you're at on their honors project, make sure we're moving along okay. And sort of related to that, I would say go ahead and start getting your projects and things in. There's no hold up. You guys can send them in anytime. And probably want to start knocking them off instead of waiting until April 14th to start knocking all 3 of them. That's gonna be a long evening. So you guys can go ahead and do that. And I'll be updating grades as you guys send them in. I'll also be able to give you guys a bit of an assessment as we head into exam 3, sort of, based on sort of how we're doing, that those grade ranges for like A's, B's, and C's are gonna change. In fact, they're gonna change in your favor. So if, for example, if you've been scoring higher than the average on the exams, you're probably already decently in the A range. So don't freak out on your grades quite yet. But I'll touch a little bit more on that next class, or maybe the class after, and we'll continue the conversation. Question 25 in the exam. Oh yeah, and you can go ahead and, if you have any questions about that, feel free to send along a question via the form, I would say. I can try and answer that in just a little bit, but I just want to get through the content for today. So I'm gonna go ahead and share the screen. Okay, so we're gonna get into the conversation. We're talking a little bit about, of course, antiviral drugs, and we were just entering sort of the the topic of proteases. Okay, so important to note, right, that even though right, as viruses pass their genetic material right over to another cell, right, it's gonna the RNA is going to be translated right into protein, but then it relies on proteases actually to chop up the corresponding long polypeptide chain to create the enzymes that are going to go through and sort of facilitate the replication process. And so these protease inhibitors have become, of course, really, really popular. We're going to talk a bit about sort of the HIV protease enzyme. This is another really fascinating area, not only in terms of sort of the, how the protein works, but also sort of how we go about designing inhibitors for it. And so the HIV protease enzyme, right, is an example from the aspartoprotease family, right? So it's going to rely heavily on aspartate groups in order to do the hydrolysis or break apart peptide bonds. And yeah, so ultimately it is a peptidase, right? So it breaks apart the peptide bonds. Symmetrical dimer that's made of two identical protein units. Active site is at the interface. And so you have the we'll talk about this in a couple slides, but you have the two sort of aspartate residues that are going to facilitate the hydrolysis, but then sort of it is C2 symmetrical. So it's actually sort of like this really neat symmetrical protein. And so what people have done is started taking advantage of that symmetry in order to design protease inhibitors. And so, kind of giving, you get a look of sort of what the interactions look like, in the binding region of a peptide, right? So you see sort of all the residues on sort of the top and the bottom. And then right in the middle, you have these, you have the two residues that are going to coordinate to the water molecule that's going to do the hydrolysis. And then you have the two aspartates in the bottom that are going to help facilitate that process. And so, once again, it's going to go through, our favorite intermediate, tetrahedral intermediate, and you have the situation where you have the two aspartates, 25 and 25 prime. So you're going to see 25, and then you see 25 prime. So that means it's just sort of the other sort of side, right? So it's symmetrical. It's got the two symmetrical sides. And so it ultimately goes to the tetrahedral intermediate, and then ultimately you end up with hydrolysis. And so design of protease inhibitors looks very much like something that we talked about before, where you have this amide here, and you can't necessarily design a drug that's going to have the tetrahedral intermediate, or that's going to fall apart, right? So what people will do is they'll sort of delete one of those OHs, right, from the tetrahedral intermediate, and the design compounds that have replaced sort of that nitrogen with a carbon or nitrogen potential, or different substituted nitrogen, and then have the OH there, right? So you have these hydroxyethylene has been a really common one. Keep in mind that when you do that, you've now introduced stereochemistry. So it becomes important whether or not you design something that's, for example, R or S. And then some other good sort of options for that is dihydroxyethylene units. It's been used pretty frequently. Reduced amide is another really good one. But what I would say is overall, the sort of dihydroxyethylene has been a really popular one, and we'll get talking about that throughout this session. So Sequinavir is sort of one of the more staple sort of protease inhibitors that was developed by Roche, and has actually truly sort of inspired the development of a majority of the protease inhibitors that have been developed since. People have turned actually to Roche to sort of facilitate the design of some of theirs. So Merck and Pfizer, among others, have even sort of come together to develop them, and that's not really as common as you think. So really neat. But as you can see, right, they use that sort of hydroxyethylamine unit, right? So this would be where the tetrahedral intermediate typically would be, right? So this would be sort of the carbonyl, and then this would be the nitrogen typically. But these molecules here, right, they start to bind remarkably well. And we'll talk a little bit about sort of once you have sort of the core unit where you have sort of that isoster that's going to function in place of the tetrahedral intermediate, you're then going to sort of build out from there to start favoring binding to other parts of the symmetrical enzyme. And so, yeah, so this is sort of what it looks like, right? So this would be sort of the bond that would break between this alpha-phenylalanine and L-proline. So this would be the transition state isoster. And what you could see then is then they continue to build out from there, accessing new sort of some binding areas. So what you're going to see throughout this, and as you can see, the development continues all the way down to sort of this bottom one that ends up with sort of an IC50 of 0.4 nanomolar. Really great. But what they do is they sort of, as they build out, they do sort of one sort of binding region at a time. So they'll say, okay, can we get something that binds to, they sort of label them, right, S1, S2 prime, etc. And so they'll have like a complete SAR series that goes into sort of optimizing for S1, and then they'll go to the next one, S2 prime, etc., which is really, really neat. And these continue to be designed exactly the same way today, which I think is really, really neat. So then Abbott sort of took advantage of those symmetrical properties a bit ago. In the active site, C2 symmetric, so the substrate can bind right left to right or vice versa. So they actually think about sort of an actual like left to right or right to left type of binding for these. And so they started to develop new ones that basically what they could do is they have this sort of tetrahedral intermediate right on the left. And so what they figured is if you just take something like that and let's say you build in the left side and then you want to make that symmetrical, what you're going to do is you're going to just essentially flip that over across sort of the x-axis and which are the y-axis, I guess. And then and what you're going to do is you're going to end up with sort of the symmetrical side on the right side. Right, and at that point, right, then this what would be a hydrate isn't stable, so they take that just to be the corresponding alcohol. Okay, so now you've designed symmetry in the entire molecule, which is really neat. I should say, while that was sort of Abbott's finding, they did consult Roche early in that. So actually, if you see a lot of these drugs, there's sort of the prefix to those has sort of the Roche and Abbott names affiliated with them. It's really, really cool. So ultimately, they see that the benzo group is really good for sort of what would be sort of the local environment where the actual hydrolysis is typically occurring on a peptide, and then once again, right, they can build out from there. Right, so they're able to get X-ray structures of these, which I think is really, really neat. And actually, those X-ray structures actually have the water bound. That's really, really cool. And so basically, what they can see is sort of exactly sort of the type of, or they're going to get a feel for sort of what the transition state looks like, when you have the compound bound. And what that does actually is informs them for elucidating sort of the mechanism of action for the proteases. It's really neat. Right. So then this concept has been extended into creating now what would be sort of the diol variant of sort of the monoalcohol compounds, right? So again, so you have this sort of reactive intermediate, tetrahedral intermediate. And what they've done is now sort of they just slide that over just a tad right into this bond right here. And what they can do is they can map this on in a similar fashion to what they did before to create ritonavir. And so what they have, right, once again, is you have these two these two hydrates now that just can get converted into corresponding diol. And so that was another whole area of development. And so what they found actually since just recently, I think in the last like 2 years or something like that, they have actually extended this out to sort of more alcohols out, and they've reached their limit. So what is cool though is the new one that's supposed to be coming out from Pfizer actually has these 2 alcohols, but then there's one more alcohol in the middle. So 3 is sort of the limit, and then 4 just doesn't bind. So that one should be on the market shortly. But as you can see, right, what they do is they do take sort of a lot of inspiration from sort of a typical peptide that would bind, right? So these are still sort of peptidic in nature. And so what they'll do is they'll go ahead and start with a polypeptide. Usually these are easiest to synthesize as well. There's actually like automated peptide synthesizers, which are super great. And what they'll do is they'll continue to make modifications sort of on the left and the right side, right? Very similar to what we saw before. And ultimately they're able to get to a compound that's sort of ideal for them. So the same game, sort of in further development. These ones actually have sort of ran out, I think, in the patents, right? So they'll start to continue to make modifications to sort of introduce, let's say, the next phase of protease inhibitors. And so what's neat about this is they already have, this is like they actually, I was talking a little bit to, again, I keep bringing this up, but anyway, the folks at the CDC, and what they'll actually do is they'll already have like 2 to 3 drugs backed up for use as soon as one runs out. So they're already sitting like a good 5 to 10 years ahead, which I think was amazing. Like this one up top was already like in development before the one before ran out. Indinavir developed sort of there was a sort of hybrid strategy developed by Merck. This is headed by Merck, but Roche and Abbott also contributed to this. This is like one area that all of them sort of get together on and talk about, which I think is really fascinating. And so they took advantage of the symmetric active site of the protease, right, yet again, and set it on a hybrid protease inhibitor strategy, where they take sort of half of what was sequinavir and another lead compound that they had from before. And as long as they have sort of symmetry sort of in sort of what would be the space that sort of usually gets hydrolyzed, then they can sort of build out with something that looks pretty similar in sort of symmetry. So what they're able to do, right, as you can see, right, if you map this, the compound on the top right into what would be sort of their abbreviated binding site, you have sort of it sort of sits nicely into each of the subunits of the protease enzyme. Niflunavir is another example. And so, as you can see, right, a lot of these are very similar practices, but nevertheless really interesting. I think one thing that's been of key interest now is actually to come up with ones of sort of understanding what types of sort of heterocycles are going to be really effective for binding in each of those sites. There's one that's in development that's pretty cool. I think it actually is sort of it's like macrocyclic in nature. So it actually, but it's symmetric across the board. So it actually if they look at sort of like molecular dynamics of it, it actually like spins instead of just binds. It actually spins like constantly in the enzyme. I think it's pretty cool. But anyway, that's just fun chemistry stuff. Yep. So Pelinavir, right, is another good example. But you can see right across the board, a lot of these are relying on that same sort of isoster, right? That hydroxymethylamine. This is by far, right, going to be the most prevalent one. Molnupiravir is another good option. I think it's worth, as you think about studying for exam 3 or just looking into it for your own interests, it's worth you sort of putting back the AMID into that position and just kind of getting a feel for, what that transition state maybe looks like. The other thing to consider is now you're gonna be jumping into sort of higher level questions as you go into your next exam. So I might ask about sort of, for example, what's if let's say you were to replace this nitrogen with a carbon, why is that going to be less susceptible to, let's say, hydrolysis in the sort of native peptide form? So when it sits as a peptide. So something to think about, maybe revisit this and in review. Happy to talk about that. Right. So inhibitors of sort of other targets, right? There's these antisense agents that are developed to block the production of the HIV protein that's necessary for the transcription of other HIV genes. I would say that this is not as popular, but one of the examples is tricoviricin. And of course, I'm likely not going to ask you about this, but at least wanted to make you aware. Yeah, fusion inhibitors are becoming a much more important thing. And so I wouldn't worry too much about sort of the sort of necessarily being GP41, it's a glycoprotein. So as that sort of fusion occurs between virus and the host cell, right, there needs to be some way to start sort of the endocytosis process, and sort of it causes sort of it involves sort of a fusion of the two membranes. And so ultimately what the inhibitors will do is actually block that fusion from occurring. And so this structure, I'm gonna have the structure of it. Oh man, I should have given you that structure. I'll send it out. And enfuvirtide actually is one that actually can bind to some of the peptides that are involved with that fusion occurring, the glycopeptides, and that can actually stop the fusion from occurring. It just functions as sort of like a steric blocker, really. Agents that limit cell entry also have become mildly popular, I would say. They inhibit the glycosidases that typically catalyze the trimming of carbohydrate moieties linked to the viral proteins. These have also been sort of interest of late. There's also, right, CCR5 chemokine receptor antagonists. Not particularly important at the moment for you guys, but definitely worth taking a look at if you're interested. Yeah, just to touch base a little bit on the flu virus. It is definitely, right, an RNA virus that infects the epithelial cells, right, so it's on the surface of the upper respiratory tract. Nucleocapsid, right, contains sort of negative sense single-stranded RNA, and the chiral enzyme RNA polymerase is important. There's two viral glycoproteins, so we'll label them NA and HA, that make up the membranous envelope surrounding the nucleocapsid. And in order to reach the epithelial host cells in the respiratory tract, it's maybe the most important, the virus has to get through a layer of this protective mucus. And so neuraminidase is a key player in that happening. So the mucosal secretions are rich in sort of these glycoproteins and glycolipids that bear sialic acid, which makes it sort of mucousy, right? So you have the situation where you have to, in order to break through that sort of mucousy membrane, you have to come up with a way that you start to detach sort of these glycoproteins from what would be sort of this sort of sugar molecule, sialic acid. And so, right, so very, very similar to what we've seen before. Once the flu virus reaches the epithelial cell, absorption takes place followed by endocytosis. You know that. RNA nucleocapsid is released in the cytoplasm of the host cell, right? It's uncovered. And then the viral RNA and RNA polymerase both enter the nucleus, and the process continues budding, right? So very similar to what we've seen, where you have sort of the absorption process. Ultimately, you get an endosome inside that's uncoated. RNA, the RNA and RNA polymerase are released, head into the nucleus, you get translation, and ultimately conversion to the nucleocapsid with the genetic material. And ultimately, that's budded out to continue the process of replication in other cells. Neuraminidase is arguably one of the heaviest studied proteins, I think, ever. And the reason for that is not only it's sort of importance in sort of the study of the flu virus, but also it's been a really fascinating enzyme to look into for people sort of across the field. So definitely a promising target for antiviral agents, as you might have imagined. It does have this mushroom-shaped tetrameric glycoprotein that's anchored to the viral membrane by a single hydrophobic sequence. It's 29 amino acids, but sialic acid is bound to the active site through a series of hydrogen bonds and ionic interactions. So, a while ago, we sort of had that picture. It was sort of one of the first ones that we identified, like sort of H bond donors and acceptors, et cetera. Very similar type of binding process here, right? So it's really, really sophisticated, right? And that you actually have this thing's like really, really tailored to binding sialic acid. I think it's really cool, right? Via all these different interactions. And so, sort of the mechanism by which sort of that breaks apart is you start with the sialic acid, it binds, and what it does is sort of facilitates the departure of this sort of OR group, that would be like your glycoprotein. And so what it ultimately does is sort of that release happens right at the sort of anomeric carbon, and ultimately generates sort of this oxonium ion, which can then be hydrolyzed just via water. And sort of that transition state sort of looks like this, gets the oxonium ion, and then that continues, from this top to here. And then ultimately what you have is sort of production of both the alpha and the beta anomer. And so the reason why that is because the oxonium ion can continuously be generated, right? Where the sugar can open and close until it has a mixture of both the alpha and beta. So transition states inhibitors mostly have been sort of replacement of that OR leaving group with something that's not going to leave. And so one good example is sort of this carboxylic acid has been one good replacement, because the other thing to keep in mind is that you also need to maintain the hydrophilicity of what you had in the when the glycoprotein was attached. And so carboxylic acid has been sort of go-to across the board for that. And so those binding interactions sort of look like this. So typically what would happen in sort of the previous mechanisms is that you'd have this sort of binding event, and then you'd have release of the glycoprotein. But now you don't have that with sort of the CO2 just there in place of it. So the polar nature of the inhibitors before was a challenge in that it wasn't particularly soluble, and it also didn't make it to places where it was supposed to. So they replaced sort of the glycerol chain with this carboxyamide, and so this is one down here, and that actually helped with sort of absorption among many other properties of the drug. Yeah, right. So then you have this situation now where you've now introduced just to, I think, to distill this down, what you have introduced by changing to that carboxyamide is you've so now introduced sort of a new sort of van der Waals interaction, but more importantly, you're able to sort of facilitate um, sort of absorption of the molecule. I think was one of the more important aspect to changing that. It also was recognized. This actually played a large role in sort of the development of Tamiflu. Um, they recognized that the dihydropyran, so the one that has the oxygen in the ring, was not necessary, and so you can increase the bioavailability by removing that oxygen. And so that was a major, super major development. So what they did is they just replaced that oxygen with this methylene isoster, so just with carbon. Um, and then they sort of introduced that double bond. They moved the double bond from sort of this position to the one up top, and that sort of helped in sort of the optimization of it. And then ultimately, again, they still had a bit of a challenge with sort of bioavailability. And what they found is sort of just using, it would be sort of an extension strategy. There's like a few papers on this. So they actually developed an extension strategy on each one of these substituents. So they put substituents on the nitrogen, on this nitrogen, and then also on the carboxylate, and didn't see much help, right, for binding or efficacy. But strangely, they came up with this what might seem a bit random of an ether group that's actually able to. It found sort of two new van der Waals interactions that sort of help in binding, which I think is really cool. It also helps with bioavailability because you've made it just a little bit greasier and it's not as polar. Cold virus is sort of another one to just touch on real quick. It is caused by a rhinovirus, and the smallest of the animal viruses. Contains positive strand RNA coated by this icosahedral shell made of 60 types of 4 distinct proteins. So this is actually fascinating in that, remember, the viruses have had to evolve over time via sort of self-assembly processes, right? So to think that you had to piece together 60 different components was pretty impressive. There's literally, in order for it to sort of invade, right, there's an attachment between the virus and the host cell that takes place in junctions between these VP1 and VP3. Those are sort of binding regions. But what's kind of cool is once it sort of parks itself on a host cell, it then relies on sort of a molecule binding in order for it to cause sort of an allosteric change in order for sort of the genetic material to be pumped into the host cell. Really sort of neat. And sort of here's like that canyon we talk about, and then you have sort of the canyon floor and the binding of the substrate to the binding region that ultimately causes the infection of the host cell. Capsid binding agents, right? They're very similar. It's the same kind of game. You sort of have these. This would be sort of the binding molecule. So you give it sort of a dummy molecule that's not going to initiate that allosteric change. And so these are a few of the ones that have been developed. I would say that Plecanaril is definitely still the most popular one, but this is another big area of development, sort of especially in the heterocycle space. A lot of different heterocycles being screened on that. And on that note, that is the end of the antiviral section. What time are we at? 10:45. Okay, so that wraps up antivirals. I'm likely going to give you a reading that sort of is going to be related to exam 3. I'd like to maybe give that to you by Friday so you guys can look it over the weekend. That sort of covers a few of these in a little more detail, but just sort of, it's more or less for your own information. I encourage you, again, if one of these like really sparks your interest to, go look into it a little bit. And if there's something that's really fascinating, feel free to send me an email. There's a lot of papers on these, but at least want to give you just a little bit of overview. On that note, I want to just talk with the graduate and honors folks for a few minutes.